This video will focus on eukaryotic chromosome mapping. Some of the major topics we'll focus on are listed on the screen there. We'll focus on linked genes, we'll use 3-point test crosses, and we'll very briefly discuss some concepts related to using molecular markers as well. So let's begin with thinking about crossing over. So you can imagine crossing over occurring during prophase 1 of meiosis. I just want to point out at this time that if you imagine chromosomes undergoing crossing over, you can actually see the physical site where crossing over can take place. And so often that's referred to either as a chiasma or a chiasmata. So again, if you come across that term, that's simply what that refers to, the actual site of crossing over taking place. Imagine a situation where we're talking about two genes, A and B, and let's say we're talking about an individual that is heterozygous for both of those genes. So big A, big B, little b. Imagine if those two genes were on separate chromosomes and therefore assort independently of each other. If this genotype here that you see, again, heterozygous for both genes, if that were in a cell that were undergoing meiosis, you can easily imagine how you can get 4 different combinations of alleles. So for example, you might get big A, big B, big A, little b, little a, big B, little a, little b. Again, just based on independent assortment of chromosomes. However, what if we now turn toward the right-hand portion of the screen here? What if the A and B genes were on the same chromosome like we see here? And furthermore, let's say the situation is that we have on one chromosome big A, big B, and on the other chromosome we have little a, little b. Now imagine if there's no crossing over between these two chromosomes and this cell undergoes meiosis, you would essentially, again, this is the same genotype, right? Big A, big B, little b, as we said toward the far left. But if this were the situation, if they're both on the same chromosome, if that cell underwent meiosis, you would just get 2 combinations of alleles. Namely, half of your gametes would be big A, big B. The other half would be little a, little b. That would be the case unless crossing over took place. And so if we focus toward the bottom, we see again the same 2 starting chromosomes that we see toward the middle of the screen here, but notice a crossing over event taking place. So if that occurs, then we would see some recombinants, namely some gametes with big A, little b, and some with little a, big B. So we're going to think about that whole process of crossing over as a way to map genes on chromosomes. Some terms you might come across, coupling conformation and repulsion conformation. If you see a situation where you tend to have the dominant alleles on one chromosome and recessives on another, that's just simply referred to as coupling conformation. On the other hand, if you tend to see dominant with recessives on the same chromosome, that's simply referred to as repulsion conformation. So some basic concepts that you want to keep in mind. So number 1, let's just go back to this previous screen here. Number 1, if you imagine 2 genes on a chromosome, the farther apart the 2 genes are, the more likely they are to have crossing over occur between them. I mean, would just be more space there, it's just more likely to occur. On the other hand, if the 2 genes are very, very close together, then that's less and less likely that crossing over will take place. Okay, so that's one key concept. The farther apart the genes, the more likelihood of crossovers. Not only that, but another basic concept I'd like you to try and keep in mind, because this will be important later on, that is when we look at double crossovers, it turns out it's the middle allele or the middle gene that gets moved between chromosomes. So that's illustrated here. So again, a double crossover, we've got not 1, but 2 crossover events taking place. And again, what you'll notice if you follow that is that it's the middle allele that gets moved between chromosomes. So you can see that illustrated here. So if we start with sort of the parental genotype, if you will, one chromosome, big A, big B, big C. The other chromosome, little a, little b, little c. Again, double crossover, we end up with big A, little b, big C, or little a, big B, little c. So again, notice it's the B allele that's been moved between the chromosomes, and that's crucial. Keep that in mind because that'll become very important later when we talk about 3-point crosses. Okay, So let's just think for a few seconds about how identifying or discovering linked genes. So first of all, if we're talking about genes that are assorting independently, so in other words, they're on different chromosomes, if you test cross a dihybrid, so we're talking two genes, heterozygous for both, that will produce offspring in a 1-to-1-to-1-to-1 ratio. So what exactly am I referring to? Well, imagine a situation here where if we just jump to the F1 generation, so we're going to cross an individual that's heterozygous for two alleles, big A, little a, big B, little b, with a tester, so heterozygous for both genes. If you do that cross, of course, the tester will always contribute recessive alleles. What you should see, and again, hopefully this logic makes sense to you, what you should see is that you would get these four combinations of alleles in the offspring, in the F2 generation, in a 1-to-1-to-1-to-1 ratio. And so if you actually did a cross like that, and you get some numbers like we see here, 255, 245, you could probably just look at those and say, yeah, those are pretty close. 1,000 total offspring, you'd expect 250 of each genotype. And of course, you could do a chi-square test just to confirm that, yes, indeed, this is close enough to that ratio. But, so again, you'd say, well, that matches the hypothesis that these two genes, A and B, assort independently of each other. On the other hand, what if you did the cross and you came up with numbers like we see toward the bottom right here? 442, 458, 46, 54. I mean, you could just look at those and say, well, that's definitely not a 1-to-1-to-1-to-1 ratio. You certainly could do a chi-square test and confirm that, but something's going on there. Well, if you have data that look like that just don't match your predicted ratio, then chances are those two genes, the A and the B genes, are probably linked because you're clearly not producing that expected 1-to-1-to-1-to-1 ratio. Some symbols that you might come across occasionally that try to indicate relationships between genes. So for example, often if we don't know for sure, or if the book doesn't want to let you know what the situation is, they might use a dot like we see toward the top here. If two genes are on different chromosomes, they might use a semicolon like we see here. If two genes are on the same chromosomes, they might use a backslash like we see here. So again, just some notation that is commonly used in your textbook. Let's now think about how to construct linkage maps. So in other words, try to get a sense of how far apart genes are on chromosomes. So let's just jump to this F1 generation right here. And let's say we're crossing an individual that's heterozygous, big A, little a, big B, little b, with a tester. Okay, so again, a tester always contributes recessive alleles. And let's say we do the cross, we end up with some numbers like we see here, right? We've seen these numbers before. What we can do, if we look at these numbers, we can recognize, again, clearly it's not a 1-to-1-to-1-to-1 ratio. Those genotypes that are the most abundant, the most frequent, those represent the parental genotypes. Relative to the parental genotypes, crossing over events are fairly rare. I mean, they occur, but they're far more rare than are the parental genotypes that we begin with. So we would say, okay, these two genotypes, the last two on the bottom, those are recombinations. Those are crossing over events that occurred. So we'd say those individuals are due to crossing over, so we call those recombinants. So what we can do is get a sense, a relative sense of how far apart these two genes are, A and B, on a chromosome, just based on their recombination frequencies. So to do this, it's fairly straightforward. You just take your total number of recombinants, so 46 plus 54, that gives you 100. Divide that by your total number of offspring, divided by 1,000, multiply 100, just to convert it to 2%, and that gives us, in this case, 10%. So we're just saying, okay, 10%, or 100 out of 1,000, of our offspring are due to recombinant crossing over events, or in other words, or are recombinants. So it turns out we could say, use that number and say, well, okay, a map unit, which is just a relative distance, is equal to a recombination frequency of 1%. So in this case, we'd say, well, genes A and B are 10 map units apart. So again, hopefully that's fairly straightforward to you. If not, certainly feel free to pause the video and just work through the numbers and make sure that makes sense. Now we're going to go a little farther and say, what if we've got 3 genes on a chromosome? In this case, let's use genes V, B, and G. And let's say we want to know 2 things. First of all, the sequence of those genes, and also the distance between those genes. How can we go about doing that? Well, we can conduct a 3-point test cross. And it's a little complex, so we'll take just a few minutes going through this particular example here. So we have, in this case, these 3 genes, V, B, and G. And let's say we don't know what the sequence of those 3 alleles is. Our goal is to figure out that sequence. Now, just to keep it straightforward, the actual sequence is VBG, but let's say we don't know that. I mean, I could have easily written this as BVG, right, a different sequence. But just to sort of try and keep it simple, let's say the actual sequence of these alleles is VBG, but we don't know that yet. Okay, so again, how does this all work? So we're going to do a 3-point test cross. So we've got a heterozygote individual for these 3 alleles. So a wild type or a dominant V and a recessive V. A wild type or a dominant B and a recessive B. And the same thing for the G. So I'm just going to hopefully keep it simplified. I'm just going to use pluses for the wild type or dominant alleles and the small letters for the recessives. So we're crossing a heterozygote with a tester. The tester, of course, is always going to contribute recessive alleles. And if you look at the offspring and the 8 different genotypes shown down here, again, notice that on the right-hand side, we essentially have recessive alleles. Again, those would always be contributed by the tester. And toward the left, we see other combinations of alleles. So let's walk through the logic here. So again, you've got numbers of individuals of each class or of each genotype toward the right here. And again, one of the first things you want to do is identify the parental genotypes. And again, those are going to be your most abundant. So if you look at these top 2, 305 and 275, those are the most abundant ones. So that indicates that these 2 genotypes represent our parental genotypes. The next step is to identify the double crossovers. Single crossover events are rare compared to the parental genotypes, but double crossover events are even more rare. I mean, they're just less likely to occur. So if we look at our individuals, the next step is to identify our double crossovers, which are going to be our least abundant among our classes. So toward the very bottom, you'll see these last 2 listed. Those represent our double crossovers, again, because those are the 2 that are least abundant. Recall earlier we said that during double crossovers, it's the middle allele that changes position between 2 chromosomes. So here's where that's relevant, because if we compare the genotype or the situation with our parental chromosomes with our double crossovers, we can compare the 2 and ask ourselves, okay, which allele is moving position? So for example, if you compare this top parental genotype, so plus, 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 with this genotype here, the second from the bottom, so plus B plus, which allele has been switched? Well, again, go back up to the parents and imagine if you flip this B and this plus, that would give you plus B plus, and that's what we have here. You can confirm that by looking at this very bottom one, V plus G. Again, go back to your parental, flip the position of the B and the middle plus. Again, that would give you V plus G. So again, by comparing double crossovers with the parentals, that indicates which of those 3 alleles is the middle one. And sure enough, we can say, oh yes, it looks like the B allele is changing position on the double crossover, so the B allele must be in the middle. Okay, again, and that's, I wrote the letters in the correct sequence, but if we didn't know, if our question was, what is the sequence of these alleles along the chromosome? Now we know. Now we know the sequence is VBG. Or you could also say GBV, same thing, right? It's just, you just flip it, but the point is we know the sequence of those 3 alleles. Okay? And again, that was our first step, and we did that by identifying our parental genotypes, our double crossovers, and finding out which allele switches position. Now, so we still have these 4 genotypes sort of toward the middle here to contend with. So now that we know our sequence, VBG, we have to identify which of these represent crossovers between the V and the B alleles, and which represent crossovers between the B and the G alleles. And again, you could use the same logic. So you could take a look at these 2 here, genotypes 3 and 4, compare those with the parental genotypes, and what you're going to see is if you flip the position of the V and this plus in the parentals, that's going to give you these 2 genotypes here, genotypes 3 and 4. Okay, so we know these 2 then represent crossovers between the V and the B alleles. By the same logic, these 2 genotypes, genotypes 5 and 6, they represent crossovers between the B and the G alleles. So now we've identified what every genotype represents. We've got our 2 parentals. These 2 are crossovers between V and B. The next 2 are crossovers between B and G. Then, of course, the bottom 2 represent our double crossovers. Now we have all the information we need. We've already established the sequence of these genes, or these alleles, along a chromosome, namely V, B, G, and we have our crossing over frequencies, or recombination frequencies, that we can use to calculate distances between the 3 alleles. So the same numbers are just reproduced toward the top here, just for reference. Toward the right, again, we've established the sequence, V, B in the middle, then G. And how do we calculate distance between those alleles? Well, again, just the same logic like we did earlier. So if we want to know the distance between V and B, we just add up all of the recombinants that represent crossovers between those 2 alleles. So again, look at our data and identify those crossovers between V and B. And so we'd say, okay, 128 plus 112. And we also have to count our double crossovers because, again, this class represents, again, a crossover between the V and B as well as another crossover, right? So count our double crossovers, so 22 plus 18 divided by our total number of offspring, in this case 1,000, multiplied by 100. That gives us 28%, or 28 map units. Do the same thing for those crossovers between the B and the G alleles. 74 plus 66 plus 22 plus 2018. Again, don't forget to include your double crossovers. That gives us 18 map units. If we were just interested in the overall distance between the 2 outer alleles, so between V and G, we could just do it in one step if we wanted to. Add up all of those crossovers that are between those 2 alleles, so illustrated toward the bottom here. And notice that you also have to include your double crossovers, but you include those twice, so you multiply them by 2. Okay. Take all of that, divide by 1,000, and as expected, right, based on the 2 individual calculations, our overall distance is 46 map units. Okay. So that's one example. That's how this 3-point test cross works. I encourage you to pause the video and maybe perhaps go through the numbers just on your own to see that indeed that whole process makes sense. Okay, the last few slides, we won't go into much detail, but I just want to point out that genes themselves can be mapped along chromosomes, but molecular markers can be mapped as well. And so just very briefly, we're going to talk about some techniques that are used to identify molecular markers that can be mapped along chromosomes. And quite often these markers, we say, represent hidden genetic variation because they are found in regions of DNA that are non-coding regions. They're not even part of genes. And so they're there. They assort in a Mendelian fashion. They assort just like normal genes do. But again, they're sort of hidden, if you will, because they aren't necessarily involved in producing any particular phenotype. So some of the examples we're going to look at: SNPs, or single nucleotide polymorphisms, VNTRs, or variable number of tandem repeats, and RFLPs, or restriction fragment length polymorphisms. So again, very, very brief introduction to these techniques. So first of all, SNPs, some background information on the side here. We're simply talking about single nucleotide differences among individuals. And so you can imagine a portion of DNA, again, may or may not be part of a gene, but a portion of DNA where most of the bases are identical among individuals, but there might be one position that can be different. So here we have either an A, a C, a G, or a T, again, depending on the individual. And so that SNP, that one base difference, that single nucleotide polymorphism, can potentially have an impact. So for example, RFLPs, or restriction fragment length polymorphisms, what do these represent? Well, these are basically situations where specific sequences of DNA are recognized and cut by restriction enzymes. Now, restriction enzymes, these are produced by bacteria. Bacteria use these to chop up viral DNA, but molecular biologists use restriction enzymes for a lot of different purposes in the lab. So if you take a piece of DNA, treat it with a restriction enzyme, that enzyme will recognize certain sequences. So in this case, looks like CCGG is the restriction site here, and it will literally cut the DNA at that site. So toward the top, if we have an individual that has this situation, so 2 restriction sites, if that DNA is cut by the enzyme, you notice we'll have 3 pieces of DNA produced: a short Y piece, a little longer piece labeled W, and a longer piece labeled X. And you can identify those by running it out on a gel, and you get something like we see to the far right. If you've got a SNP in someone else, and they just have a single base change, if that happens to be in one of these restriction sites, then that will no longer be recognized by the restriction enzyme. So cut this DNA with the same restriction enzyme, you're just going to get 2 pieces of DNA produced. A small piece here labeled Y, and a much larger piece labeled Z. And again, that's illustrated on a gel toward the right. So the point is, you can use these RFLPs sort of as markers, if you will. And these also, of course, represent differences among individuals within populations. Another marker that can be used are VNTRs and STRs, variable number of tandem repeats and short tandem repeats. So it turns out that regions of DNA can be repeated. Sometimes the regions are kind of long, maybe 100 base pairs. That's what we find in VNTRs. Or sometimes they're short, just a few bases, like CAT, CAT, CAT. And it turns out that the number of times that those sequences are repeated can vary among individuals. And so these regions, these VNTRs and STRs, can be identified simply using PCR, where you have primers on either side of your repeated units of DNA, and you can amplify those. And again, the differences in terms of how many times those sequences are repeated can be different among individuals, and again can assort just like normal genes do. So again, you can run PCR, and you can identify, for example, homozygotes and heterozygotes just based on the number of banding patterns that you get, and that identifies the approximate size of those pieces of DNA. So again, those can be used as molecular markers as well. And one of the techniques that's kind of interesting, it's called FISH, fluorescent in situ hybridization. So here we can use this technique to actually visually identify roughly where a gene is on a chromosome. So what you're looking at here is a a cell that in some stage of mitosis, so you see the chromosomes here, and it was treated with a molecular probe. So if you have a gene of interest, you know the sequence of DNA in that gene, you can create a probe, which is simply a piece of DNA that is complementary to your gene of interest. You can attach fluorescent molecules to your probe, add it to a slide with actual chromosomes like we see here, visualize it under ultraviolet light, and essentially anywhere your probe hybridizes or attaches to the DNA, again because the gene would have the complementary sequence, that region will then fluoresce. So you can literally visualize it under ultraviolet light, again to get a sense of where the gene is that you're interested in looking at. So that wraps up our brief discussion of mapping eukaryotic chromosomes.